we encourage you to perhaps order in from a local restaurant that's doing takeout or delivery and enjoy it that way. We've got a bunch of really interesting talks coming up for Origins Science Scholars starting on Tuesday, the 14th of April, going all the way through the end of May. And we've got Dustin Tyler from our biomechanical engineering department talking about the new body-machine interface. We'll have a coronavirus update in about a month. We have Nikki Burt from the Cleveland Museum of Natural History talking about mother and child health. We'll have Chris Cullis talking about climate change, fire, food, and our future climate. Bob Savinel will be talking about smart and sensible renewable energy. And Johannes Haile Selassie will be talking to us about his new discovery, newly published discovery of Australopithecus anamensis, a new skull with the face. So these will be super exciting. So anyway, without further ado, tonight's rather serious topic is understanding coronavirus. And we'll have Hirsch Mathur, professor of physics, and Neil Greenspan, professor of pathology in the medical school. We'll have Neil go first. He'll be talking about immunity to viruses. And then we'll have Hirsch talk about mathematical modeling.
If we could, just before we get into that, I just want to say from the Happy Dog side of things, thank you so much for doing this, for allowing this to go forward online and including us in this. It's such an integral part of who we are, and we really miss being able to be there for everybody, but being able to be a part of this and knowing how much Patricia, Glenn, Hirsch, Neil, and everybody at the Institute, what you've done looking out for us as part of this, and just incredibly grateful. So just wanted to say that before we got started. So thank you so much.
Well, we're grateful for the trust that you've put in us over the years to take over your bar and put science in it. So, and I love seeing that you have a hot dog there in the back. Is that with the Fruit Loops? And you've got the beer.
Yeah. Yeah, thanks, Sean. It's been great working with you, and we're looking forward to many more years of doing that.
Oh, great.
Just before Neil and Hirsch get started, just a reminder of the message on the screen. If you're not one of the speakers, I'm about to do it myself in a moment. Please turn off your video and turn off your mic. It'll make the talk go much more smoothly for everyone else. All right, here I go. Video off. And
hi, Glenn. So, all right. So tonight we have understanding coronavirus. Of course, knowledge is rapidly multiplying on that, and we hope to continue to keep you updated in the weeks to come. We'll have first Neil Greenspan and then Hirsch Mathur speaking. A couple things to know. The name of the virus the official name is SARS-CoV-2. But the name of the disease is COVID-19. So sometimes these can be confusing, but that's how it is. A quick timeline. In November of 2019, there were a few early cases of pneumonia. The virus was sequenced by some folks, but nobody knew how significant this would be. In December, there were more cases of pneumonia. By the end of the month, the World Health Organization was alerted. Into January, suddenly there were tens of thousands of cases, 250 people dead. The World Health Organization declared it a global emergency. Cases began showing up in the US. By February, there were 100,000 cases around the world, 3,500 people dead. The first US deaths. Lockdowns began in various countries. Beginning of March, the World Health Organization declared a pandemic. Ohio, just to bring this a little bit home, we had our first cases on March 12th, but Dr. Amy Acton, the director of the Department of Health for Ohio, had seen this coming from a distance. She had people already training a month before that. So we're ready to go. Ohio is one of the states that have taken action most effectively. So as of today, there are 867 confirmed cases in Ohio. 17% of them are healthcare workers, and that could be a problem. going forward. There are only 15 deaths so far. 17,000 people have been tested. The virus has shown up in at least 60 counties out of our 88 counties in Ohio. We have successfully mitigated the surge that would have been hitting right now to mid-April at least, and hopefully till May, if people keep doing what they're doing. But I think that Hirsch will talk a little bit more about that with the mathematical modeling. As of today, in the US, 75,000 cases, and we passed 1,000 deaths earlier today. So this is the virus. This is from the CDC. And I will then turn to Neil, if you want to share your screen.
Okay. So I'm clicking on the, oh, there it goes. Okay.
Got it? Okay, good.
Okay. So I'm going to talk about immunity to viruses in general and link that to our current situation with SARS-CoV-2 or novel coronavirus. So I'm going to talk about how the immune system responds to a virus or any other pathogen or antigen. An antigen just being a molecule or a complex of molecules that elicit what we call an immune response. And there are different kinds of immune responses, so-called innate immunity and so-called adaptive immunity. Both are adaptive in an evolutionary sense, but on a short time scale, there's a bit of a difference between adaptive and innate immune responses. Adaptive immune responses, which are carried out by lymphocytes primarily, and there are two kinds of lymphocytes as indicated on the slide here. There are B lymphocytes. These are the circles with little shapes that are supposed to represent receptors for these foreign or antigenic molecules, and T lymphocytes. And what makes an adaptive immune response so-called adaptive as opposed to innate is that the lymphocytes are capable of, each lymphocyte will recognize only one or a few antigens, as opposed to innate responses that tend to be more promiscuous in what they recognize. That's one way to put it anyway. So we're primarily concerned with lymphocytes, therefore, if we're going to talk about adaptive immune responses. And what an immune response fundamentally involves is that cells, B cells or T cells, that have the ability to recognize, meaning bind to, in what's called a non-covalent interaction, but essentially that's a way to latch onto a foreign substance. Those are the cells which will get so-called activated. That's the terminology that immunologists use, meaning all kinds of functional changes happen in the cell. And among those changes are a program that's encoded in the DNA of that cell to proliferate when it gets activated. And so let's say we have a B cell that's specific for the novel coronavirus, SARS-CoV-2. It has a receptor that binds the spike protein, which is the key protein for the virus to infect host cells in the lung. If a B cell with the receptor, the green one, has a receptor for the spike protein and none of these other B cells do, then this B cell will, and it's illustrated here, so let's think of this as the SARS-CoV-2 spike protein, then that B cell will become more prevalent in subsequent, at subsequent time points. And effectively, this is a Darwinian or neo-Darwinian form of somatic cell evolution. Somatic cells are different than germ cells in that they're what make up most of the body, and they can't be passed on to offspring typically. So the same kinds of things happen with T cells, but today we're going to focus more on the B cells because in the end, what I want you to understand are the products of B cells called antibodies, and the surface receptor is a form of the antibody that's bound in the membrane. And the antibodies that mediate immunity are the ones that the B cells secrete, or their progeny, so-called plasma cells, secrete into the extracellular fluid, including the blood in particular. So this is a process we call clonal selection, in that each cell or group of cells that have the exact same receptor are called a clone. and the antigen selects the clones that have the right receptors. So it's a very clever scheme in that the immune system has to respond to a vast array of different potential threats on a molecular level, and the threat is what actually guides the immune system to choose which cells to amplify. So as you can see here, the green ones get amplified more than the rest of the B cells that don't recognize the coronavirus. And so they end up being the ones to secrete a lot of antibodies. So what does an antibody look like? So here we have this color-coded scheme. This is a T cell receptor for comparison, and we're not going to spend much time on that. It's very similar. It's sort of like this portion of an antibody. Antibodies are made of two polypeptide chains. Polypeptides are also known as proteins. They're strings of amino acids. Amino acids are important elements of what we need to ingest every day to form our own proteins in our cells. Proteins include enzymes, include molecules like collagen that make up extracellular tissues. The key part of the antibody for binding to antigens is in this part here, and these two domains here are called variable domains, and the rest of the domains are constant domains. And there's a heavy chain in blue and a light chain in red. And so there's a heavy chain variable region and a light chain variable region or domain. And this is the membrane form. So you see these little blue pegs are going through the membrane. They're called transmembrane regions. So this is on the surface of the cell. This is inside the cell, and this is outside the cell. But after a B cell gets activated, because its receptor binds an antigen, like we talked about in the previous slide, it secretes into the blood antibodies, which are basically the same structure minus the transmembrane regions. And these antibodies can mediate immunity, and I'll show you how on the next slide. Now, there's quite a bit on this slide, but basically what I'm showing you is that there are multiple different mechanisms or pathways by which antibodies can help protect the body against an invading virus, bacterium, fungus, or parasite like malaria or leishmania. Okay, so among the most important diseases are bacterial diseases where toxins are secreted by the bacteria. The most famous examples are diphtheria and tetanus, which secrete what are called exotoxins. These are bacterial molecules that have very deadly effects on host cells. And if the body makes antibodies, if the B cells in you make antibodies to diphtheria toxin or tetanus toxin, the antibodies can bind to those toxins, prevent them from binding to their receptors on host cells, which is how they get into the cell and cause damage. And then the complex of the antibody and the antigen, the toxin, can be taken up by what are called phagocytes. These are cells that have a very special ability to ingest material and destroy it by digesting it. So that's one mechanism. And the same kind of process can happen with viruses. It's called neutralization. So you can neutralize toxins and you can neutralize viruses. The other mechanisms shown here are more important for bacteria, and that includes binding directly to the bacteria by the antibodies, and that can lead to uptake by phagocytes through some molecules on the phagocyte surface that recognize this portion of the antibody, the back portion, far from the binding site for antigen. And then another mechanism involves activating proteins in the plasma called complement proteins that also interact with different receptors on the surface of the phagocyte, and they can synergize with the receptors that recognize the antibody. And the complement receptors and the antibody Fc receptors, so-called, can facilitate uptake of the bacteria and destruction. So those are the different kinds of mechanisms that are routine. There are some other ones I won't go into, but these are the main ones that immunologists and physicians worry about. So for our purposes, neutralization is not the only mechanism by which antibodies can protect against viruses, but it's one of the more important ones. Now, the novel coronavirus, as it was being called for a short time, or now SARS-CoV-2, has this protein called the spike protein. And the spike protein is what allows the virus to attach to a molecule on the cell surface of a host cell, like an epithelial cell in the lungs. And the particular receptor on the lung cell that's made by the human cell is angiotensin converting enzyme 2. And the original SARS virus uses the same receptor, as does the new coronavirus. So they're very similar basic structures between the two different viruses, but they're not that cross-reactive at the level of antibodies, meaning that antibodies to the original SARS virus spike protein will not necessarily bind effectively to the SARS-CoV-2, and antibodies to the SARS-CoV-2 spike protein will not necessarily bind to the spike protein on the SARS molecule. And actually, you can see here, this is the novel coronavirus protein in green, and here's the original SARS coronavirus spike protein, and they're similar, but they're not identical. So there's a rough structural similarity, but they're clearly different, and the amino acid sequences are different enough that antibodies will recognize most of the time only one or the other. There may be some cross-reactive ones, but to a large extent, the immunity is not overlapping. That's at least what we've been led to believe so far. So where does this leave us in terms of applications to our current situation, where we're most concerned with, as Hirsch will get to, delaying the surge of cases so that we don't overwhelm our healthcare system. And it will also do our healthcare system a great deal of good to have more time, because in the meantime, we can develop better testing or more testing. We can develop perhaps therapeutic agents. We may be able to develop as well immunological therapies or vaccines. And the vaccine approach would involve essentially what I showed at the very beginning of this brief talk, which is an immunogen derived from the virus, most likely the spike protein. And there was a trial done with the SARS virus spike protein, the original SARS virus, 2002, 2003. This was done some years later, but it didn't go anywhere because the threat had evaporated by that point. But they did show in a small trial at the NIH that the spike protein could be used as a vaccine, and in 10 patients, 9 of them responded quite well with antibody and T-cell responses, and it seemed to be safe. A very small trial, but it was at least proof of principle. So what I'm talking about on this slide are a couple different approaches where we could use antibodies to protect people. Either one possibility would be to treat very sick individuals who are already suffering from lung disease from the SARS-CoV-2 infection, COVID-19 disease. And there are a couple of different ways of doing what's called passive immunization. It's called passive immunization because you're taking antibodies formed in someone else and transferring them. You can inject them potentially intramuscularly or intravenously into a patient who's, let's say, in the hospital, who you're trying to prevent from going into acute respiratory distress syndrome. So you can get antibodies out of convalescing patients who have recovered from the coronavirus infection, from COVID-19, who are a certain number of days out, at least a couple of weeks out, who have titers of virus, which in vitro are shown to be neutralizing. And the FDA, just 2 days ago, approved an emergency new drug application to try convalescent serum therapy for very sick patients on a patient-by-patient basis. That's one approach. The other approach, involving much more modern technology, is to create what are called monoclonal antibodies, wherein you take a particular B cell and the genes it uses to make antibodies, just that one B cell, effectively. And you can do this with multiple B cells in parallel, potentially. And you generate an antibody from that one B cell that you've screened and shown can neutralize the virus or mediate immunity to the virus. Then you can produce huge quantities of this very homogeneous product. The difference between that and a serum from someone is that it's a much better defined product, and you could potentially make unlimited amounts of it, as opposed to convalescent sera, which are unique for each patient, and you can't go back to that patient and expect the serum to be the same as time passes. And anyway, so the convalescent serum idea is probably going to be tried very, very shortly. There are companies already working on making monoclonal antibodies that can potentially mediate immunity to the virus, although all of these points need to be verified by properly done trials as well as in vitro experiments. But there's an even more modern approach, which I think does have some particular interest in terms of some of the issues that we'll be facing as we go forward in this pandemic. And that's what's called vectored immunoprophylaxis. Pretty long word, but it's pretty simple if you break it down. So vector just means that we're going to take a gene that encodes the antibody, or genes, and we're going to put them in a virus that doesn't cause disease, but is effective at getting the genes into a host cell, like the muscle cells. And so you can take a virus like adeno-associated virus and put in an antibody, insert an antibody into the genome for that virus that encodes an antibody of interest that is specific for the coronavirus, spike protein, for instance. We can inject that virus into your muscle, into your upper arm, let's say, and then your muscle cells will become little factories, if you will, to make antibody. Now, they won't have all the same properties as B cells, but if they spit out enough antibody, as long as that gene works to make messenger RNA, which then gets translated into protein, you would have antibody in the circulation that could protect you or treat an ongoing infection. So you could use this potentially, probably more for prevention as an alternative to a vaccine, which might be interesting in a group like older people who are the most susceptible to serious disease because they often have weakened immune systems and don't mount good responses to vaccines. So for those people whose immune systems have declined for whatever reason, who are less likely to respond well to a vaccine, this is a really interesting possible avenue of protection. So with that, let me just move to my last couple of slides so that Hirsch can begin to get ready. And I want to cover some of the important issues which we'd like to know more about, but given we're only a few months into this pandemic, needless to say, much more research needs to be done to be absolutely definitive about answers to these questions. So the first is, does infection with the SARS-CoV-2 reliably induce immunity to repeat infection? We don't know that yet because we haven't gotten to the point where there's a risk of repeat infection. If we get to the point where this pandemic declines and the number of new cases drops to almost none or none at all, and we have a period where it's pretty quiescent, and then it starts infecting people again, we'll begin to see if people who had prior exposure are protected, which is, in fact, the origin of the term immunity, which is actually based on people who are immune from Roman taxes, but it got applied to the immune situation with pathogens. If we do reliably induce immunity with infection, how long does it last? That's going to be also important in terms of whether this will turn out to be a seasonal virus or not, and what kind of herd immunity we get. We want to know, can complete protection against infection be provided by immune mechanisms? We're hoping so, because there are certainly viruses where antibodies can provide complete protection from infection. But it's not true for every virus. And so that needs to be nailed down with actual experiments and data and observations. And then we want to know, is protection mediated primarily by humoral immunity, primarily by cell-mediated immunity, which means T cells, T lymphocytes, or are both types of immunity required? And then there are cases with other viruses where one or both are critical. Generally, antibodies are critical for preventing infection, but sometimes cell-mediated immunity is critically important for recovery, even if it can't absolutely prevent infection. And then we also want to know if recovery, same point I already kind of alluded to in the last blurb, does recovery result primarily from humoral or cell-mediated immunity or a combination? And then there's one concern we have, How did that happen? Try again.
Could be your computer's saying you're out of time.
Oh, that could be. So the final slide, whoop. I'll just do it in this mode. The final slide, there's a phenomenon which has been seen with some viruses like dengue, where antibodies for the virus, a different serotype of the virus than the one that's circulating at the moment, can actually enhance infection of host cells. It's called ADE, or antibody-dependent enhancement. And we don't know if that happens with SARS-CoV-2 antibodies, but we need to know that and find out pretty quickly, because we don't want to be putting antibodies or inducing antibodies with a vaccine, which is active immunization, if it's just going to make the infection worse. And then we want to know if antibodies in the sera of recovered COVID-19, or so-called convalescent sera, COVID-19 patients, are able to prevent initial infection or treat ongoing infection. And that, as I said, that's going to be subjected to some small individual patient trials very shortly. And then we can try the same issues, address the same issues with monoclonal antibodies, or cocktails of monoclonal antibodies, which might be more effective. And again, we can do the same with vectored immunoprophylaxis, particularly for prevention, but maybe even for therapy, if that turns out to be a possibility. So really, that's all I wanted to cover, is just to give you an overall sense of how the immune system responds to viruses, and how, using antibodies, we can potentially treat or prevent infection. And we can move on to Hirsch or take questions, however Patricia wishes to handle it.
How about if you take your screen off and Hirsch can put his up, and while he's doing that, we'll take a quick question or two.
Okay.
If someone has a question, just unmute your mic and just ask the question.
Hi, this is Maureen, and thanks so much, Neil, for stepping forward to do this. Really appreciate it. So my question is this. So we've been reading about these things that are called cytokine storms in individuals who seem to be truly suffering, and maybe this is even associated with the people who actually pass away from this disorder. So what is that? Is that some sort of a manifestation of an overimmune, an uber immune response?
Yes, so it's not at all uncommon that the pathology associated with viral or other pathogen-mediated infections is caused in part by the immune response. And genetic variation in patients will influence the extent to which that is a problem. Now, of course, many pathogens can cause direct host cell damage and tissue damage by releasing toxins and by killing cells through other means. But there are many cases where, toxic shock syndrome is a classic case of that, where the Staphylococcus or streptococcal bacteria release toxins that activate T cells regardless of their antigen specificity. They're called superantigens, which is, I guess, a term one could quibble with. But the point is, they do activate a lot of cells, more than a typical antigen would. And the result is secretion of cytokines to a degree which is excessive. And that, in fact, it is basically a cytokine storm. So what's going on with the coronavirus, it appears in many patients, is excessive inflammation in the lung. And clearly this isn't happening in every patient. And part of it may be that people with pre-existing, particularly lung morbidity, but also cardiovascular and kidney problems can apparently predispose people to being more susceptible to this pathway. So it's definitely the case that the host response is often part of the problem. And I think the virus may cause some damage to the lung epithelium directly, but it appears that much of the damage that leads to acute respiratory distress syndrome is being caused by the cytokine response. And so that would involve, in addition to adaptive immune mechanisms, what many people would refer to as innate immune mechanisms, which can also generate a lot of cytokine production. So, you know, the whole picture is more complex than I covered in just talking about adaptive immunity with a focus on the B lymphocytes. But T cells can secrete cytokines as well as B cells, but T cells probably secrete more. Then there are other cell types that are
Hey,
part.
Neil, I think we're going to need to leave it there.
Okay.
There's so much to talk about, but
No, it's a huge topic.
it's a huge topic. That's why we're going to come back to coronavirus from time to time in coming weeks so that people can learn a little by little. Yeah. So thank you so much. And Neil will be around for questions after Hirsch's talk as well. All right, take it away, Hirsch.
Okay, great. Well, thanks, Patricia, for organizing this. And also thanks, Sean, for hosting us at the Happy Dog. So I should say, by way of my own background, I'm interested in models of infectious disease and the broader subject of dynamical systems, both from a teaching and research point of view, but actively working on modeling coronavirus. So in effect, I'm providing a kind of outsider perspective. But what I'm hoping is that this talk will help you make sense of the headlines, and maybe if you feel drawn to it, to start reading the primary literature on your own as well. Now, there's a rule that Stephen Hawking enunciated in his popular book, Brief History of Time. He said that every time you put an equation into a popular book, you lose half your readers. So I've violated that rule. There's a few equations in here, but I want to make it very clear up front that this talk is intended for people in the audience who disavow any knowledge of algebra or calculus. If that's where you are, that's exactly who I'm speaking to. Nonetheless, I have included some equations because I feel like if you're going to be reading the primary literature, you need to see what they look like and how to walk around them. Okay, so what is mathematical biology? Well, the eminent mathematical biologist, Martin Nowak, tells this story about a biologist who was hiking in the mountains, and he comes upon this really bored shepherd, and the shepherd says, if you can guess how many sheep are in my flock to within, say, 10, you can have one of them. So the biologist says, sure, you have 180. And the shepherd is astounded, and he says, you're right. And so the biologist picks up an animal, the mathematical biologist, I should say, picks up an animal, and the shepherd, wait, if I guess your profession, will you give me back my animal? Biologist says, sure. What are the odds the shepherd has heard of mathematical biology? And the shepherd says, you're a mathematical biologist. So he's stunned. How did you know? And he says, well, that's because the animal you picked up is my dog. So I guess that's obviously just a rather self-deprecating joke, but it makes I guess the takeaway from the story is that they're obviously good people because they have a self-deprecating sense of humor. And in this case, I think the work they're doing is, of course, of incredible importance to us right now. So I want to start by talking about exponentials. Of course, it's become a colloquialism to talk about the exponential, about exponential growth and so on. But I really want us to feel this and get a gut feeling about how explosive exponential growth really is. So let's imagine that there's a hypothetical infection which has infected. We're going to do a simulation. And the good thing is that we can do the simulation without a computer. We can do it right here. So we're going to imagine that there's a infection, there's 100 people who are infected, and the number of infected people double every 3 days. That's the situation on day 0. Let's see how quickly this infection grows. So 3 days later, double the number, 200 are infected. Well, that's not a lot, 200 people. Day 6, almost a week has gone by, it's still only 400, maybe not a big deal. Day 9, it's 800. Day 15, it's 3,200. So 3,000 people. Well, that's a lot of infections, but it's compared to the population of the country, maybe not that much. So let's carry on. We go on to day 30 now, continuing to double every day. And there's actually a shortcut I didn't find print. So now we've got 100,000 infections at the end of a month, just doing this doubling thing. And if we keep going to 2 months, we've got 194 million infections. That's basically 2/3 of the US population just in 2 months, starting from 100. So that's the power of the exponential. Where does this come from? Well, there's a very nice graphic in the New York Times, which was work of these epidemiologists. I just realized I don't have a pointer, but I guess we'll make do without. So what you can see in this picture is how the exponential comes about. You have this one person in red, and he infects 2 people, say within 3 days, and then each one of them infect. Thank you. Annotate maybe, and mouse. No, that wasn't the right choice. Spotlights. Oh no, well, it's okay. I think we'll try and get by without. I'm still not able to activate it. Oh, annotate. No. All right. Well, I think we'll be fine. So each person infects 2 more, and they keep doing it in 3 days, and you have this explosion, this rapid growth of infection. Actually, the same diagram could be used to illustrate how a nuclear chain reaction happens in a nuclear explosion, like in a nuclear bomb, which is certainly known to be a very explosive thing. And another nice thing we can learn from this picture is, in this case, each person is infecting. That's called the reproductive ratio. And in principle, of course, there's no reason why it needs to be 2. You could be infecting three more people or fewer 2. And this is a very important parameter in determining how the infection is going to spread. Okay. So this picture also allows us very nicely to see how early action can prevent the spread of infection, because if just that one connection was missed, half the people in this picture are not infected. And so that's one of the few good things you can say about an exponential, that if you start taking action early, you can actually make some inroads. So of course, the real infection doesn't keep growing exponentially, because pretty soon there are a number of factors that cause it to ultimately start slowing down. It may start exponentially to begin with, but it'll reach a peak, and then it'll even decline. So that's what you see in these pictures over here. They show how the infection would progress without protective measures. That's the sharply peaked curve. And then how you can flatten out that curve. And I'm sure you've seen this hundreds of times, that if you can somehow slow down the infections, basically by going back to the strategy of trying to keep infected people apart, if you can slow down the infection, you don't actually necessarily reduce the number of total infections, but you certainly slow them down. And that, as Neil was telling us, is really so important. If you only have 10 beds in your hospital, you don't want 100 patients showing up. So if you can decrease the number of, the rate at which these cases happen, if you can lower that peak, that makes a huge difference. If you can delay that peak, you might be able to develop some of these technologies, again, that Neil was talking about. And so that's why it's so important. Now, this picture is just a passive still picture. But in the New York Times, just today, in fact, one of the op-ed columnists, Christoph, he published an interactive model in which you can see how this progression of the disease depends on interventions that we might take, or on assumptions we make about the virus itself, about how deadly it is, what its reproductive ratio is, how long it incubates the infection, and so forth. So you can play around with all those parameters, and you can see how these curves shift. So what I want to do is to try and talk a little bit about those models, of which this op-ed contains a very simple example, what ingredients go into those models, and what we can learn from them. There's a number of important parameters that characterize these infections. The reproduction ratio is one of them, which we already saw illustrated before. This has to be bigger than 1, otherwise the infection is just going to go away. But if it's bigger than 1, it begins to grow. And this parameter is really a measure of how infectious the disease is. So you'll see a lot of talk about this R, or R naught is usually how it's denoted. And for the flu, for example, our is 1.5, but for COVID, it's higher, it's 2.5. And that's what makes COVID more dangerous than the flu. There is really no comparison. Another important parameter is the case fatality rate, which means exactly what you might think. It's what fraction of the infection die. And as you can see, we've had a couple of other related epidemics in this century, in the 21st century. There's SARS, which was 10% fatal, and then there was MERS more recently, which was 35%. The seasonal flu is just 0.1. Ebola, that's actually the worst Ebola outbreak, was 90%. More often, something like 50%. Now, COVID is, is above the flu. It's between, we don't actually know the number yet, but we think it's somewhere between 0.3 to 1%. But it's definitely a lot less deadly than, let's say, Ebola. And the take home from this is that, you shouldn't be complacent, because the effectiveness or the deadliness of an infection isn't really just measured an individual fatality rate at all. For example, if it's too dangerous, the disease doesn't propagate because people die before they can infect others. And that's the situation with Ebola. Not that we should underestimate Ebola, I'm not advocating that at all, but I'm just pointing out that this number can be very misleading. Sometimes you see a small number and you think, oh, we're safe. I'll show you another graphic, in fact, to illustrate that in just a moment. And another important parameter, if you're trying to figure out policy, of course, is what fraction of the infected people need hospitalization. And that's what creates a crisis with COVID as well, because the number not known yet, but a much larger fraction of the infected will need hospitalization and critical care than the flu. And this is why COVID can overwhelm our healthcare system, whereas the flu, it's something that happens every year and we have capacity. I'm not saying everything is ideal, but it's not going to overwhelm the system in the same way that COVID can really do. So here's another graphic showing many of the things that we've just discussed. But it also puts a number of diseases on the vertical axis. It's the fatality rate. And as you can see, COVID is not very high on this. Ebola is like on top, and COVID is just a little bit above the seasonal flu. But it's also not very far. In fact, it's above the great 1918 pandemic, which killed somewhere between, well, at least 20 million people in 1918. And that's been the most deadly pandemic of the 20th century. So again, you see this case fatality rate should not lull you into a sense of complacency. Okay, so now I want to look under the hood and say a little bit more about the models. So here's where some of the equations begin. First of all, let's write down a really simple model, one that leads to exponential growth. This is of course not realistic because at some point it's going to stop growing exponentially. The take home from this is that in order to get exponential growth, or what makes exponential growth happen, is when the rate of new infections increases in proportion to how many infections you've got. That's what that equation is. But again, if you're averting your eyes from the equations, what I want you to carry away is that the simplest model that I'm describing, it has only one variable, which is the number of infections we have. And it has one parameter, this thing called beta, which basically determines what the doubling time is. So the distinction I want you to understand is between the variables in the model, which in this case is the number of infections, and the parameters that get put in. And the way we figure out what these parameters are is that we go out and we count up the number of people who are infected, and then we plot that against time and we try to figure out what the parameters are as best we can. There's lots of uncertainties because we simply don't know the number of infections, but we try to fit as well as we can. Now, there's a missing figure here. Next up in complexity is a model that acknowledges that ultimately the exponential growth has to stop, because if everybody is infected, there's nobody left to infect. So what this model does is it assumes that the rate of new infections grows in proportion to the number of people who are infected, which is n, the number of infections, but it's also in proportion to how many people are not infected, which is n minus n. So that makes sense because the way infections spread is when the infected meet the uninfected. So the rate should depend on both those things. And that's a feature we'll find in more realistic models also as we go along. This model has two what are called fixed points by mathematicians. If no one is infected, then the rate of new infections is 0. And of course, that makes sense, right? If no one's infected, no one's going to get infected, and we're all fine. And the other fixed point is when everybody is infected, then again, the rate of new infections is 0. And the troubling thing about this model is that if you're in between those two, if you have some people infected, you're not at either extreme, it inexorably takes you to the bad extreme, the one where everyone's infected. But that's just a model, it's not reality. Now let's look at something that's a little bit more realistic. And here's a model of infection, the simplest model one can write down for infection. Actually, before I go on to the infectious model, let me just talk about the happy situation where there is no disease. This is a disease-free world. And in this case, there's only one variable, which is the population. And then there are two parameters, which is the birth rate, because people are being born, and then there's the death rate. And the death rate, or the rate at which people, some fraction of people years. So that's proportional to the number of people there are as well. That's what these equations are saying. A model like this always ends up with a steady equilibrium population. If you wait long enough, the population will always take this value k over u, which is called x star. So no matter where you start, this is a very happy, healthy world. You always converge to a stable population. Now let's bring disease into this world. So what's going to happen is that there are now two variables. There's the healthy population, the number of healthy people, that's x. And there's another variable, which is the number of infected people. And this yellow term over here, that's the term that's the rate at which people are becoming sick. And that's proportional to how many healthy people you have and how many sick people you have, because again, it's their interactions that cause infection. And then this term V, that's the death rate due to sickness. And so you see that the infected people are dying off faster. The death rate for them is U plus V, whereas for the people who are healthy, it's just U, the normal death rate. What happens in a model like this is, well, it really depends on all the parameters of the model. You might end up with the infection dying out, and you might end up at the healthy fixed point we talked about before with the population of X star. Or you might end up with a situation where the disease is permanently embedded in this population. And if you wait long enough, you end up with a smaller population of healthy people, and you end up with a steady population of sick people as well within this model. And the number of healthy people, it's down by a factor of R, which is just the reproduction ratio. And it depends on the parameters of the model in this way. So there's a lot of equations here. This is the last and most complicated slide I have. What's the take home here? The take home again is that if there's a infection, the rate of the infection is going to depend on the interactions between the healthy and the sick people. And that's why interventions that minimize those interactions can be effective in slowing down the infection. That's one takeaway. And I guess, yeah, that is the main takeaway here. All right. So now let's move on to models that might actually describe coronavirus. And the simplest models you can write down, and there's a very nice model. Yes, there's a very nice model of this sort, which was created by Alison Hill at Harvard. What she's done is she's created, it's a standard model, standard kind of model, but what she's done is she's created a simulation which you can go off and run. It's very much like the simulation in Christoph's op-ed, but it gives you much more insight. You can look under the hood and you can play around with the parameters and really, really explore what's going on. So I would encourage you all to do that. You can find it just by Googling, I guess, her name, and or I'd be happy to send you a link. In this kind of model, you've got a number of variables. You've got the number of people who are exposed. These are people who are asymptomatic. They're not infectious, but they do have the disease. And then you have the infect to various degrees, mild, severe, and critical. And then you have the population of people who've recovered. And then, of course, you have the population of people who died. And then there's a lot of parameters that describe how interactions between these different populations cause them to move from one population to the other. If you're a susceptible person and you meet an exposed person, then you'll become infected and so on. And so the parameters that describe these different rates. So in this model, in the simulation, which you can access online, you can actually play with these parameters and see what effect they have. You can also introduce interventions. Basically, the interventions go towards changing some of these parameters. One of the things I'd like you to notice is that these parameters, none of them are actually the ones that you read about in the news. They're not the case fatality rate or reproductive ratio. Those are things that emerge in terms of these more raw parameters, which you actually measure from whatever data we have about the progression of the infection. And so if we have good data, we can figure out these parameters, we can run the models, and we can try to predict the future as well as we can. We can also try to analyze the past, about which we might not know very much. Oh, and it says acknowledge Mike Inchevsky. So I should say that Mike pointed me to this simulation that you can run online. And indeed, I'd hoped he would be the one giving this talk. I hope he'll be back in the future to tell you more about this. So what are the kinds of things people are trying to figure out by running some of these models? Well, some of the papers I've been looking at recently, there's this paper that appeared in Science, which asks the really important question. We all know that we don't have enough tests available right now. And for example, in Ohio, we know that there are 500 cases, but there might be many people who have the infection and we simply don't have tests and haven't had them tested. So what's the true number of people who are actually infected? Can we somehow estimate that based on the number of known infections? And so there are models that attempt to do that because this has happened before. We can track the data in China and try to correlate these things. So if you do feel inspired by this article to read the literature, one of the things that will strike you is how tentative everything is right now, and how people are working to try and arrive, how many things are unknown, and how people are working hard to arrive at the answers. The other thing, if you're not a regular reader of the scientific literature, that'll strike you is how bad they are at catchy headlines. Lee et al. Probably thought this was real catchy. Substantial and undocumented infection facilitates the rapid dissemination. But the New York Times, describing the same work, called it coronavirus is hiding in plain sight, meaning that there are many more infected people that we don't know about than the number that have been already tested. Another interesting paper, and this one, whereas the previous one was in a prestigious journal, this is on something called medRxiv, where this is what researchers do nowadays. You finish some piece of work, you don't wait for the publication process, you post it so that other people can benefit from it right away. And so this paper tries to address the question of asymptomatic patients. Are they infectious? That's a really important question, obviously. If you're showing no symptoms, can you still infect people? And again, we're trying to address this question through models because it affects the progression depending on what the answer to is. So you can play around with and see if it makes the model fits the data better if you're using people who are asymptomatic. And here's another interesting paper, still also on archives, still in preprint format. And it asks the question, when did the outbreak start in northern Italy? And so we know that the first documented case was mid-February, but according to this model, perhaps it was really in early January, and that would explain why interventions have been so unsuccessful, because they already had thousands of cases and many weeks had gone by. Now, I should emphasize that these are very preliminary results, and people are debating these things, these ongoing discussions. And that will apply equally to the article that was published in the prestigious journal as much as the ones that are preprints. These are just questions people are asking, but these are questions that models will eventually and hopefully soon help us answer, because we need to understand things like how long this had been festering, and to understand what didn't work and what works. There's a vast literature now already in this field. So Nature made a graph, and there's more than 1,000 publications. Very hard to keep up with all this literature, of course, but then there are specialized subfields. And okay, so I guess I'm out of time. One of the things everybody is doing these days is offering advice on what you should be reading or watching. And so I'm going to do the same. People usually say Camus. Maybe I'd say Camus as well. Read Camus. But more on the topic, there's this interesting document that the CDC produced 2 years ago. If you read it, it reads like a prophecy. It was there, because they've been preparing for this for years and decades. And every time there's an epidemic, they refine their models and their strategies. So the expert advice we are lucky to be getting from the scientists, from the CDC, it's founded on decades of research. And you can see that 2 years ago, they were already anticipating and advising on, giving pretty much the advice we're getting now. And then this other report from Imperial College is, at least according to the newspapers, it's been enormously influential to the extent that science is informing policy. It's had an enormous impact, and these are both really, really good to read. All right, I'm going to stop here, and I'm happy to take further questions.
Harsh?
Yeah.
You said that the reproductive ratio for COVID was 2.5.
Right.
How did they come up with that number?
Well, so if you go back to a model like the one I was describing, the innards of that, the one that you can simulate on the shape of this curve depends on all the input parameters. And so from the data we have, you can try to fit, the parameters. And then from that, you can calculate the reproductive ratio.
Okay.
I should say there's a great deal of uncertainty, though, of course, about everything.
Yeah.
So, the 2.5 number comes with error bars.
Yep.
Hey,
Right.
Hirsch,
Comment on that.
this is Sean. Kind of a layman's question. Is there modeling to identify which geographies are more susceptible based on the populations in those particular geographies and pre-existing conditions and
Right. Absolutely, yes. So there are indeed models that, and I should have said this is a very important point. All the models I described didn't really worry about, the fact that there is such a thing as geography, right? They were just talking about the number of infected people. And so what are we talking about here? The number of infected people in Ohio, in Cleveland, the whole world. And these models don't have that built in. So that's a huge limitation. I was describing these models because they're easy to describe and because you can run them easily enough on, on a laptop like the one I'm using right now to speak. And those are usually the things that interest me the most. But if you want to do a real good model, and some of the literature I was pointing to afterwards, all the serious modeling, all the papers that I point to, they do have geography built into them. And so they take these things into account. And indeed, yes, those models can be used to address all these important questions like what effect does climate have? And, is there a seasonality to this and so forth? As far as I know, we don't actually have any definitive answers, although, and I certainly haven't seen any papers that, claim to have anything very definitive. But I do know that the CDC very recently, either yesterday or today, I was reading in the news, say that they have some basis to believe that there's seasonality, but I haven't seen the scientific analysis on which that's based.
Hi, Harsh. I have a question. First of all, thank you for the really interesting talk. So I was wondering, in China, where the virus originated, it seems like they've seen a positive trajectory with the virus, where the vast majority of people have recovered already, and they were reporting very few new cases. So why can't we expect a similar trend to occur here and in Italy and around the world? And were there certain measures that they took that allowed for that steep decline relatively quickly?
Yes, that's a very good question. So I want to go back to the graph. And what they have managed to do is to flatten the curve, I guess. And they have managed to, although they had a huge peak. And so, probably if they'd started earlier, they might have been able to produce a better outcome as well. But they're definitely over the hump. And, at some point we will all be, the question just is, which hump are we going to go over? Are we going to have protective measures and manage to keep within our healthcare capacity, or are we going to shoot over that? In terms of, have they been more successful than we have, or are there lessons we could learn from them? The two countries that seem to have it, seem to have managed to, got to a point where they don't have further infections, South Korea and China. People point to both of those. Are somewhat different strategies in the two cases, but yes, it's working, and so there's certainly a lot to learn from that.
Okay, cool. Thanks.
Virtually everybody else I know of is in that exponential growth phase, and that's just such a terrifying place to be right now.
Right.
I just want to make a quick comment about the R naught.
Mm-hmm.
It's often talked about like it's an intrinsic property of the virus, but as you basically noted, there are a lot of variables which will affect that average transmission risk. And, if you have an incredibly densely populated city like Wuhan versus, some rural area, there probably is an intrinsic transmissibility you could look at under some defined conditions for different viruses.
Right.
But how densely populated an area is, what kinds of congregations take place. It also will depend, of course, on which is part of the virus's attributes, what kind of transmissions are possible. There may be more than one kind of transmission, and the mix of what mechanisms of transmission are operative could probably vary in different circumstances. So I've generally seen that everyone believes that droplets, respiratory droplets, are the main mode of transmission for SARS-CoV-2, but I don't know that aerosol is 100% ruled out, or even fecal oral transmission or fomite-mediated transmission. They're probably secondary or even not important, but there might be places where they become more important. So it's an interesting, it's an incredibly important variable, but it's problematic if it's seen as an absolutely intrinsic property of the virus that doesn't vary.
Yes.
Of course, it can vary with their mutations.
Yeah. So I'd really like to chime in and underscore that as well. R is not an intrinsic property of the virus, and it's also something that's under our control to some degree, right? Because if we can reduce infections by social distancing, we can reduce the value of R, and that's why I sort of jump back to my slide with the simple model of infectious disease. You can see over here on the right-hand side at the bottom right, you can see how the reproductive ratio depends on the parameters, that raw parameters that go into the model. And it depends on this parameter B, which you can see is that term in yellow. This is the rate at which infections are happening because people are infecting each other. This is where we can come in. We can change B by reducing the contact between the infected and the healthy population. And as you can see, the reproductive ratio R depends on this parameter. That's totally under our control. Well, not totally under our control with extraordinary hardship, but it is something that is not intrinsic to the virus. It's about human society. And so I should have said that as well in the earlier question when I was asked about the R value of 2.5, that represents some kind of global average. It's maybe not necessarily a very meaningful thing. We really should look at it country by country, and then we should be asking about, what it is effectively right now and what policy can do to mitigate it.
So let me we have the great good luck that we have Dr. Deborah Lindell from the Francis Payne Bolton School of Nursing with us. And I wondered if, Hirsch, if you could put the curve back up, and if she could just talk for a minute about the practical aspects of, what does this mean when we look at it in real terms, in practical day-to-day terms. So Dr. Lindell, could you just say a few things about that?
Sure. Thanks.
You can put your video on if you would.
Okay.
There we go.
Yes, from a clinical aspect and the blogs that I'm reading, being written by various nursing organizations and position statements coming out from nursing organizations. The dotted line that is on this graphic is so critical because the flattening of the curve allows us to try and manage within the capacities that we have or that we can build as quickly as possible. And that is already being strained, as you well know, from what we're seeing on TV, but I'm reading it in blogs and all that relate to nursing and healthcare. And so this whole idea of staying at home and all the things we can do to try and reduce the spread takes those models and puts them in to real life. And already in New York City, they're saying that they think it may be helping, although they are severely strained. We have students at one of the big medical centers in New York City, doctoral students, and I talked to one today who runs 2 floors of operating rooms in one of the biggest medical centers there, and she's been having to convert them into critical care units. And the biggest challenge is trying to find staff that the OR nurses aren't prepared to care for those patients, so they're trying to find staff to do that. So this plays out in real life. The other thing I was going to mention is that I teach in Wuhan, China. I have for many years, and just this past week, I've been teaching a course by Zoom, ironically, on advanced community health nursing and epidemiology. And the ones that are in the faculty that are in Wuhan have been in quarantine for over 60 days at this point. And they still have at least another week or two, to probably almost 2 weeks to go. And their system has been very strained too, and they've responded. But that dotted line and that flattened curve are real life.
Thank you so much.
A question. Presumably, one of the challenges is that you from these, and that you can see in these models, that you take an action to reduce the transmissivity, and it's going to take a couple of weeks to show up. Is that the case?
So you're asking if we sort of introduce social distancing now, is there going to be a time lag before the curve?
Before we see the effects.
Yeah, that's a good question. I think that's got to be true. Yeah. But these are sufficiently complex that you really just have to run the model.
But presumably that's part of the challenge in places like Italy and the US, is people are used to getting results quickly. But in this case, if it takes a couple of weeks for infections to manifest themselves, if you socialize yourself, you're not see the decline in the growth of the epidemic for that lag period.
Yeah.
So you have
I think that's perfectly correct. And you can actually already see it from even this picture over here, that the 2 curves, the deviation between them grows as time goes by. So in the beginning, you don't see that much difference. Of course, over here, they're vastly different parameters. Like one is totally out of control and the other is very well controlled. But if you change the degree of control slowly, I think it will be true, as you were saying, that you would see the effects. It would take a little bit of time to see them. And so you need patience and an attention span to see those things.
Great, thanks.
So I think we've gone over our hour, which was what we had planned. Are there any pressing questions here that anyone has at the end here?
Harsh, you mentioned reading Camus for reading material. The first thing that came to my mind was Kafka's Metamorphosis. Because I feel someday like that character who wakes up as a cockroach, like everything has changed, and yet I'm trying to go through my life day to day like nothing's happening. It's a kind of strange feeling.
Yeah, I agree with that. I feel that particularly today. I hadn't really thought of that before, but yeah, I've recorded a week's worth of this morning, and so it's not life as we know it. This is the way we live now.
Patricia, can we turn on our microphones and clap?
That's it. That's a very good idea, I think.
Thanks everyone for coming.
Thanks, and we look forward to welcoming you to our future Life, the Universe, and Hot Dogs and Origin Science Scholars and all the other programs that Siegal is offering, Siegal Lifelong Learning is offering as well. So
Patricia,
stay well. And
Patricia, will you send out a regular announcement about all of these?
Yes, I'll send it out to our OSS list and our fellows list, but also all of these are going to be listed on the Facebook, and I will try to get them up on the ISO website as well, which is Origins.case.edu.
Okay, thank you.
Yep. All right, well, thank you all so much, and we'll see you
thank
next
you, Hirsch. Thank you, Neil. Thank you, Patricia. Thank you, Deborah.
Yep. Thanks, Patricia, Glenn, and Sean, and Hirsch. That was great.
Thank you. Yeah.
Yeah.
Thanks, everybody.
Thanks everyone for coming. Yeah.
All right, bye.
Good night.
Bye.